Welcome, Mindsetter Grade 10s. Finally, your time has arrived. It's Life Science Wednesday, and Kathy's with us.
Hi, guys.
Guys, in last week's lesson, we looked at animal cells and compared these with plant cells. And for the next 2 weeks, we'll be looking at cell division. So stay stuck to those seats and tune in, and we'll be telling you everything you need to know. Guys, tune in. Start posting comments on Facebook. That's facebook.com/learnextra. My name's Indiana, and I'm sitting behind Twitter as well. Our Twitter handle is @learnextra. What we're going to do is we're going to take a short little break, and then we're going to get straight onto the lesson. See you now. Hi guys, and welcome back. I know that you're all ready for today's life science lesson, and Kathy cannot wait to get started. I can see the hands going already. So guys, you know what? Kathy, take it away.
Thank you, Indy.
It's a pleasure, Kathy.
Hi, everybody. I hope you remember everything we've done so far. But today we're going on to mitosis, which is cell division. Now, you need to know the difference, and if you understand the difference now, you don't have to worry about grade 11, you don't have to worry about grade 12, because this is how cells are made. All right, now think logically. If, you know when you've had a crack in your lip, when you know your lips get very dry and it cracks, or you have a broken nail and the nail grows beautifully and you can file it nicely, etc. Or you do something and it rubs some of the skin off your hands, what happens? New skin cells form. Now how? Why? Where? When? Because of cell division. All right? The cells next to the damaged cells, first of all, the damaged cells get cleaned away. All right, and the bits and pieces, and then the cell next to the hole or the damaged cell, which is now gone, you've got like this vacant spot. So that cell makes a duplicate of itself so that we now have two cells. And that is what mitosis is. And the way to remember the difference in meiosis, which you are going to do in grade 11, in grade 12, you're going to do meiosis. That's for sexual reproduction. So you halve the chromosomes. So when you take daddy's sperm and mommy's egg and you put the two together, you have a whole baby. All right, so you've got to halve it so that when they join, you have a whole. But for mitosis, which is also a form of cell division, okay, just think, when my toes grow, my shoes get too small for me. So if we look at cell division, first thing we're going to have is this. This is the whole process of cell division. And if we look here, you can go to www.onlinetutor.eagetutor.com. All right? Indy will put this onto the web, at least onto the Facebook for you, but this is how cell division happens. These are the different phases. And as we go to those phases, you'll understand why everything happens the way it does. So, before we go to the phases, I want to show you something. Okay, we have cell division. All right. What's going to happen is the cells are going to make more of themselves. So, with cell division, we have mitosis. And this is what you are going to be studying. Mitosis is for the following processes: for growth, for repair, for replacement, and then for reproduction. But what kind of reproduction? In unicellular organisms. All right, now let's look at each one of these. All right, Just remember, mitosis, when my toes grow, my shoes get too small for me. All right, you are always going to have the duplication of one cell is going to duplicate itself. So you're going to end up with two identical cells, and we call them daughter cells. So you'll end up with two identical daughter cells. Why? Why does mitosis happen? Why does this kind of cell division happen? Because we need to grow. Now, you have grown. Think about it. We give birth to a little baby. A baby, anything from normal is about 3.2 kilos to 4.5 kilos. That's little, all right? And look at you. So you were a little baby once, and you have grown. Once you have reached your full height, your full growth, especially you boys out there, you are going to, if you're watching this, you're anything between 15 and 17 years old. You are going through the biggest growth spurt of your life other than when you were 2. All right? Remember, and believe it or not, a 2-year-old at 24 months, that's for boys, and at 22 months for girls. If you measure that 2-year-old and you double it, that is their height as an adult. So in the first 2 years of your life, you grew, if you were boys, the first 24 months of your life, you grew to half the height you are now. That's a lot of growing. And girls at 22 months, because us girls, we all, Indy, don't you agree? We're far more developed, we're more mature. We are XX chromosomes, you know?
Yeah.
Okay, so poor you boys carried on growing to 24 months. Us girls at 22 months, we had reached our height, and if you double that, that is how tall you are. Now, I know you're all saying, talking nonsense. Go and find a 2-year-old or a 24-month-old little boy, and if everything is normal and there aren't any growth problems or nutrition problems, you take that normal 2-year-old, I promise you now, they will come to here. Believe it or not. You double that height, it's within a centimeter or 2 what the adult height will be. So that's growth. But even once we've reached full growth, our nails continue to grow, our skin continues to grow because the Malpighian layer keeps making more epithelium or epithelial tissue, all right? So it keeps making more of it. Your hair continues to grow, your eyelashes. Guys, the hair on your faces that you keep shaving, all those things carry on growing, your toenails. So we continue to grow. And believe it or not, your nose and your ears as well. That's why old people, babies always look like they have very big heads, and old people always look, especially old guys, always look like they've got bigger ears and bigger noses, because those, I mean, I'm not talking about not hobbit.
Slightly bigger.
Yeah, just slightly bigger, but they grow slowly, but they continue to grow. All right, so that's growth. Then repair. Now, clearly, we have things that break in our body. Cells get mauled, they get messed up. We slough off 5-6 layers of skin a day, all right? That's what the bed mites live on, although it's a disgusting thing, think of bed mites in your bed, but that's what they live off, is the dead skin pieces, all right? So repair, replacement, all right? So if a cell is hurt or it's damaged, and it has to be replaced, it's cleaned away and a new cell takes its place. So replacement, and then, now look at this, it's reproduction in unicellular. Unicellular means one cell. So unicellular organisms, if we have lots of cells, we say it is multicellular. We are multicellular. But if an animal, or at least an organism, only has one cell, hello, it can't have a reproductive structure, okay? It can't do all those things. It's just one little cell. So if it wants to duplicate itself or make more of itself, it must therefore undergo a process of mitosis, all right? And we call that binary fission, which you will do at a later stage, but it means to make another of that cell. So this is mitosis. And if you think, for example, a plant, also undergoes mitosis. I mean, I've been talking about animals all the time, but plants as well, because plants continue to grow for their entire life. They grow, 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 grow. They make seeds, and the seeds grow, and the fruit grows, and everything grows. And then if it's an annual plant, it dies at the end of its season. So, but if you think of the roots grow down into the soil. So the growing tip of a root is called the meristematic region. And if you look at a bud, you know, the buds at the end of branches, also where little buds grow for new little side branches to develop, you are going to have your apical bud there, and it's going to be the meristematic tissue. It's the tissue that makes new cells. It's continually undergoing mitosis for growth, repair, replacement, and reproduction in unicellular organisms. In other words, organisms that are only one cell big. So you know when you get a bacterial infection, and what happens there? They're unicellular. So one bacteria makes two. Each of those two now make two, so I've now got 4. Each of those 4 duplicates, so we now have 8. And we go 1, 2, 4, 8, 16, 32, 64, 128. And before you know it, within a couple of hours, you have a fully fledged bacterial infection. Why? Because each little cell just keeps doubling and keeps doubling and keeps doubling. And that is growth. All right, so it's growing. It's making more and more and more of the population. How? Because it is unicellular reproduction. Okay, so that's mitosis. Now, let's look at our phases. How does it happen? Now, people, when you are going to have a party, what takes the longest time about the party? It's preparation. It's sitting, deciding, who am I going to invite? So you write your list of the people you want to invite. Then you decide to put it onto BBM or iMessage or
Facebook,
Twitter, or you Twitter, and you send out your invitations. You normally do it 3-4 weeks in advance. Why? Because it's a special occasion. It's your birthday. So you make sure that no one else can book that date because that's the date you've decided on. Then you go and your mom and your dad and you decide what you're going to do at your party, what you're going to eat, what you're going to drink, where it's going to be, what your cake's going to look like, what you are going to wear, especially if you're a girl, it's what the nail's going to look like, the jewelry, the whole bang shoot. And you plan for 3 to 4 weeks. And then the day of the party arrives and everybody rushes around, and then the party lasts for 3 or 4 hours. Maybe 5, maybe 6 hours tops, all right? You've had your party, but you've spent 4 weeks preparing for that party. Now that is the same with mitosis, and this is how it happens. What you've got is this part here, and at this part where the cycle begins, the cell starts to prepare. So it first of all grows in size. So there's my brand new little cell. And it grows inside. So G1 is the period of growth. So I'm going to write here physical growth. So the little cell is now physically growing and it is working. So it is functioning. Okay? It's doing what it does and what it needs to do. Then we go into this R phase, and as we get into that R phase, which is just the last end, it now starts to prepare. And this process here is preparation. And that preparation phase here, so it's now grown, now the prep starts. And here, the cell, first of all, gets all its chemicals. So, you know what? We're going to duplicate the chromosomes, because you've got to make sure that every single organelle in the cell is doubled up. But the biggest, most important part of the cell is the nucleus, because you've learned that the nucleus controls all cell functions. You also learned that the nucleus controls cell division. And you also have learned that it's because of the nucleus that the hereditary characteristics in the chromosomes, in the nucleus, are passed on to the next cell. All right, so what have we got? We've got all this prep, and we've got to like get all the stuff into the cell so that that cell can duplicate and make sure that there are copies of the chromosomes. All right, so this is the preparatory stage, and it carries on, and we've now duplicated. In fact, all of this really is prep, and now we go into the G2 phase, and here we have the duplication. Or we can say the copying of the chromosomes. So now that's done, okay? And what do we have after that? Let me just take, no man. All right, this little part here, this little piece here, there, that is where cell division takes place. So literally, your G1 is the growth phase, and then from here onwards, it's preparing for cell division. It's like you, when you have your party, you have this preparation, and then you have your party. And that is mitosis. Why are we doing mitosis? Come on. For growth, for repair, for replacement, and for reproduction in unicellular organisms. All right. Now, trick question. Would a virus be classified as a unicellular organism? And I need for you to think about that, and I want you to post your answers onto Facebook. And Indiana's going to read them when we have our little ad break just now, and give me feedback on what you said. But think very carefully. Is a virus, will a virus undergo mitosis? In other words, is it a unicellular structure? I've already told you a bacteria is. All right, so, and yeast, same thing. Also unicellular, reproduction by mitosis. And what do we call that mitosis? Binary fission. All right, so here is the cycle, and we've got the growth, we've got the preparation, getting all the chemicals, the duplication or copying of the chromosomes, and then everything is ready for cell division to take place. All right, now, let me just get my next slide, and I actually want a blank, so I'm going to go here. Okay, when we look at mitosis, there are 5 phases or steps. I'm going to say steps. And of those 5 steps, 4 steps, 4 steps are classified as phases. Because the first step is interphase, which is step 1. And inter, you know, means between. So it literally tells you it is the between phase. So it's the phase where all the preparation takes place. And that was that whole G1 and the S, and we go to G2, and then we have mitosis happening. So all of that we say is interphase. It's between the phases, so it's not actually a phase, and it's where the cell is preparing chemically for cell division to take place. All right, so phase 1, not really a phase, but it's the phase in between. So it is interphase. And remember, inter is between. Intra is inside. Okay, and I think we're ready for an ad break here, so I'm going to hand you over to Indy.
Guys, we are ready for an ad break. I think Kathy wishes she could just take it to ad break herself. Maybe next time I'll let her. Guys, thank you so much. You guys are obviously loving the show. I just wanted to quickly read out a comment from Clementine. Said, well, cool stuff, learn extra. I think everyone in my class should be here. Clementine, I agree. Tell them all to switch channels now and watch Mindset Learn. Guys, we'll see you now after the break. Hi guys, and welcome back. Guys, thank you so much for posting on our page. We've got a few answers that I'm going to read out to Kathy now, and Kathy's going to tell you why your answer may be wrong and maybe why it might be right. Oh,
okay, I think I'm going to come here with my umbilical cord.
Yes,
Kathy.
We've got
all these wires dangling all over the show.
Okay, so Kathy can have a proper little look at everyone's answers.
And do you know I need my glasses, darling? And that really gives me age. But okay, but just read them to me.
Okay, this one's from Koketso. She said, I think a virus does go through...
Koketso's gorgeous.
I know, she's
You're
beautiful.
Gorgeous, my hun. Totally gorgeous. Okay.
Yeah, I know, there's some beauties on here. I think a virus does go through mitosis because it also multiplies itself. Reproduce the cells, isn't it? Thinking.
Okay, now listen, people. A virus is not classified as a living structure. In fact, there's a lot of argument between the scientists because there are 5 groups of organisms, and viruses just has its own grouping, literally. Why? Because it does not have a metabolism, so it's not classified as a real cell. What you have is viruses that attack animal cells will have DNA, one strand of DNA, and viruses that attack plant cells, because plants are a little doffer than us, you see, plant cells only has an RNA strand. So what they need in order to reproduce is a host. So what happens is the virus will invade a host cell. It then comes down and it sort of encapsulates it, and it sits there and it has a little drill, and it drills into the cell membrane, and it drops its RNA if it's a plant, or DNA if it's an animal cell, drops its DNA into there. And then that DNA will use the cell's nutrients and chemicals to make new little viruses. So a virus itself is incapable of any form of metabolism. It cannot reproduce. So you have to look at cells that are a complete cell, and that's your smallest cell, or your smallest grouping will be, for example, your bacteria, your phytoplankton. Those are one little cell structures, and they are able to undergo mitosis. But viruses, not. And that's why we can't kill them. That's why antibiotics don't work on viruses. Okay.
Okay, perfect.
There you go. So for those of you that said no, viruses can't undergo mitosis, perfect. They can't because they're not living. They don't have a metabolism. They only really become alive when they go into a host. And remember, viruses can crystallize. There's nothing else that can crystallize and then come back to life. So there isn't much life there.
You can read that out, Kathy.
What's that? Oh, that's Lloyd Lolo Patsika.
Read it out.
Oh, is that?
Thought so, Kathy.
Yeah, thought so, Kathy. Just was confused. Okay, cool.
They get it. They're
and
getting
then Kagiso, it is not a unicellular organism. Oh my goodness gracious me, look how gorgeous Lolewa is. Sure, you gorgeous little girl.
I know.
All right.
Don't worry that you're late. Don't worry that you're late.
Yeah. Our gorgeous girls. We should actually go and have a look at how many really... Guys, you have no idea. Go look at those stunning girls on that. And girls, go have a look at the stunning guys. All right, so here we go. We're talking about mitosis. There are 5 phases, although the first phase is not really a phase. It is the in-between phase. It is interphase. Now, the second phase, people, you've got to know this off by heart. The second phase is what we call prophase. Okay? And then your third one is metaphase, which stands to reason, because if there are 5 steps or 5 phases and the first one isn't really a phase, meta means middle. Okay, so whenever you see the word meta, it always means middle. It's the middle phase. Then 4, let's just make this go up a little bit. For 4, we have Anaphase. And then 5, we have Telophase. All right, you may have a textbook that says Telophase, in other words, there's an E here, but it's the difference between American textbooks and English textbooks, all right, or your British type textbooks. It's the difference between American English and Oxford English, if you want to put it that way. They are both correct. So I like telophase. There we go. So telophase is the last phase. It's the end phase.
Kathy, we have a mindsetter named Rafense, and she's put quite a great little thing onto the Facebook page. She said a great way to remember the phases of mitosis is by this. I don't know how she would say it, but this is how I would say it. I would say IP-MAT, all in I, P, and then M-A-T.
IP-MAT. I'm getting there. Getting there.
Oh,
IP-MAT.
Rafense, she knows all about IP-MAT.
There we go. IP-MAT. It's the way to remember it, and if you remember that, I promise you now, you know it forever, and you have to know it for grade 11, and you have to know it definitely for grade 12. All right, because in grade 12 you're going to do Mitosis, which is for sexual reproduction, and you have to be able to explain the difference and the similarities between mitosis and meiosis. So just learn it now. So mitosis, why? For growth, for repair, for replacement, and for reproduction in unicellular organisms, and that does not include viruses because they are not really classified as living organisms. All right, so we've got interphase. It's the between phase. It is the preparation phase. It is also the longest phase. All right, it takes up between 80 and 90% of the time is here, your interphase. Now we go to a prophase, metaphase is in the middle, anaphase, and then telophase is at the end. What do we want? We want to make new cells. So telophase will look like the shape of an 8, all right? Why? Because we now have 2 identical daughter cells forming. That's the end phase. That's telophase. Metaphase, everything is arranged in the middle, so that's why it's called metaphase. And interphase, you are not going to be able to see anything different in the cell. Why? It's the phase that's in between. So interphase, IP-MAT, your first phase. So if we look at all of these, we go I, P, M, A, T. So that is your I, P, M, A, T. And we say it is IP-MAT. But there are actually only really 4 phases, which is prophase, metaphase, anaphase, telophase. Why? Because interphase is between the mitotic divisions. All right. So interphase, between, you can see nothing. Prophase, metaphase, everything's in the middle. Anaphase, telophase, everything is it's now making 2 separate cells. That would be in an animal. In a plant, it will do this. You'll have the cell wall starting to grow across to divide it into 2 different cells. Okay, so those are your processes. Now, how do we tell the difference between prophase and anaphase? Well, interphase, you can see nothing. Prophase, your chromosomes have now gone from a chromatin network, which looks like that, to actual chromosomes. All right? You can see them, you can identify them, and they're all mushed up within the nucleus. Anaphase, everything is pulling towards the poles. So you've now got your chromosomes heading in the opposite direction. All right? And you've got this nice big space in the middle. So that's anaphase, prophase, everything is scattered around. And if you remember this, and you remember it is ip-mat, interphase, prophase, metaphase in the middle, anaphase, and telophase at the end. All right. Now, the way I remember it is that telophase, when do you telephone your friends? When do you call them? Is when something's happened. It's when it's over. Then you phone your friends. So telophase, you're going to telephone them at the end. So telophase always in the end, metaphase in the middle, and interphase the beginning. Now the problem is pro and anaphase. Prophase, everything's just a mess, mixed up, and anaphase, everything is organized. All right, so let's move on. And here we go to our interphase. Now, if we look at interphase, this is the first phase. Why? It is the prep phase. Okay? And it is the longest phase. Remember, when you're having a party, to prep takes the longest. The party itself goes quick, quick. All right, what have we got? We've got a cell. We are looking at a cell here, but the nuclear, here's the cell membrane, okay? And the nucleus is enlarged. Because can you imagine if we try to draw mitosis in a cell that has a Golgi apparatus and smooth ER and rough ER and mitochondria all over the show, and if it is a plant cell, a couple of plastids, so leucoplast and a big vacuole, you would get so confused you wouldn't know what you're doing. So what we've done is, since we're not interested in all the other organelles in the cell, we literally take the nucleus and we focus only on the nucleus. It does not mean that all the organelles just disappear. They don't just like go into nothing, but we are focusing, when we do our diagrams, we focus on the nucleus because the nucleus is large. Why? Because it's about to undergo mitosis. It's going to duplicate itself. So we have the cell surface, so the cell membrane is here. So we know it's a plant cell. An animal cell. How else do we know it's an animal cell? First of all, it's got this round shape, but there are some round plant cells, but it only has a cell membrane. There's no cell wall. That's number one. Number two, do you remember, and I just want to get, no, hang on, sorry, I get pink. Let's go with pink here. Do you remember these little guys? Okay, now where did we have these little guys? It is your centrosome. And if you recall, where plant cells had plastids, animal cells had a centrosome. And what was the function of the centrosome? It was for cell division to produce spindle fibers. Okay, so in plant cells, we are going to have the centrosome, which splits. Now, in this one, we've got two. We shouldn't have two. We should only have one. And each one of these little loggy things looks like that. And then you've got another one at right angles. This whole thing is a centrosome. Okay, but each one of these is a centriole. All right, now what's going to happen is they're going to separate out, and as we go through interphase and move towards our next phase, which remember was IPMAT or ipmat, we're going to go to prophase. As we are ready to go into prophase, these centrioles are now going to move away from each other, and we're going to start with little spindles starting to develop from them. Right? So we've got that. So your centrioles tells us immediately, hello, we are looking at an animal cell. Right, we're looking at an animal cell. If you look at the cell and there is a cell membrane and there is a cell wall and it has a very straight, rigid surface, right, and there are no centrioles, and you think, it's a plant cell. And what they may do in an exam is your teacher may give you a plant cell because it means that if you understand it, you can apply all of this there as well. So we've got our centrioles, we've got the nucleolus. Now I wonder how many of you can remember the function of the nucleolus as a organelle. The nucleolus's job, and I'm going to do this in white, so there's no, so you don't make any mistakes. The nucleolus is for protein synthesis. Why? Because the nucleolus makes sure that the little ribosomes are made, okay? And that the RNA is where it's supposed to be and that it's being produced so that protein synthesis can take place. So the nucleolus literally provides the tools for protein synthesis to take place. So that's why we say it's important for protein synthesis. We have the chromatin threads. Now if you look here, the word chromatin should tell you it is to do with the chromosomes. And if it's to do with the chromosomes, it means it has to do with heredity characteristics. So it's for what you are going to inherit. All right. And remember, we want an identical cell. We literally, it's like making cupcakes. We want, or taking a cookie and making different cookies, we want that shape to be exactly the same. It must be an identical cell. You can't have a liver cell flapping around here on your skin or on your lip. Can you imagine, I don't know, a photoreceptor in your eye, and well, your lip's cracked, so guess what? You get a whole row of photoreceptors on your lip. I mean, or maybe even a cardiac cell, so it can go and you think, oh my gosh, what on earth is going on here? So we want an identical cell to replace the cell that has been damaged or that needs to be repaired, or that if we're going to grow, I want nail cells to grow here. I certainly don't want lip cells to grow here, okay, or heart cells to grow here. And that is why mitosis is so important. So we've got the chromosomes, and they're all mushed up, and they're all stretched out, and they're like spaghetti. And then we get the nuclear envelope, which is literally the nuclear pore that forms an envelope around to hold the chromosomes here and the nucleolus in place. All right? And I'm going to hand over to Indy.
Okay, guys, I hope you're really enjoying the show. Can you believe we've got about 15 minutes left? So if there is anything that you are wondering that Kathy's been talking about, please post your questions on the page. We're going to take those questions in the last 8 minutes of the show. I'm also going to put up all of these diagrams that Kathy's been talking about onto the Facebook page right now, and that way it's pretty cool because you can just download it onto your cell phone. You've always got it there, and you can just check, interface, need to check it out, and you know. Guys, it's so easy. I'm telling you, as long as you stick with us, you'll know everything by the end of the year. I'm going to take a quick break, and we'll be back with you soon. See you now. Hi guys, and welcome back. I hope you're all enjoying the show. We've only got a few minutes left. Guys, get those questions in. If there's anything you don't understand, please let us know. Hopefully it's all quite straightforward. And don't forget that I am posting the diagrams on the Facebook page for you guys right now. So check them out, download them, share them, let your friends know. And right now, let's get back to Kathy.
Okay, boys and girls, very easy. EPMAT, interphase, not really a phase, it's a between phase. It's the longest phase. That's what we've got here. It's the preparatory phase, is where the cell gets ready chemically to duplicate, to divide. And I really don't like cell division because it sort of gives you the idea that the cell sort of is cut in half, like an orange being cut in half. It's actually not. The cell makes, if it was an orange, it would make a duplicate orange. All right, so longest phase, first phase, interphase. Inter, between. All right, you have a nuclear envelope. It's your nuclear membrane, and the reason we call it an envelope is because by the time we finish the next phase, all we're focusing on is that nucleus. So it's the nuclear envelope because it envelops the processes that are going to be taking place, okay? Your chromatin threads, the mush of chromosomes, your nucleolus for protein synthesis, your centrioles, which the two together form the centrosome, your cytoplasm in the cell, and the cell surface membrane. And please remember what I said to you. Doesn't mean the organelles just sort of disappear and go somewhere else. They're all there, but we're not focusing on them. We are focusing on the nucleus, which does get bigger and it does enlarge. All right, now on to P for prophase. Okay, here we go. Now, prophase, remember I said to you, how will you tell the difference? Well, if we go back to interphase, you can see nothing here. This looks like a normal cell with a normal nucleus. Now, we go on to prophase, and whoa, hang on, something's sort of like happening here. Aster, and aster is a flower. All right, it's actually a very pretty yellow flower, and you get them in orange as well. Aster, what are we sitting with flower labels here? Well, the aster, if you look here, let's get a nice pretty color. You've got sort of like the little rays coming out here. The aster is what we are going to have developing into spindle fibers. So those are the little fibers that are now going to be coming from the centrioles. Now you'll see here, we say centriole, there is no S, because this line is pointing to only one of these little structures here, okay? And let's do it in pink. So there we go, all right? That's one centriole, this is the other centriole, and they are at right angles. Now this here would be early prophase. It's the beginning of prophase. And if you go to university and you do microbiology and that kind of thing, you will do 8 different phases of prophase, 8 different steps in prophase, 6 different steps in metaphase. We're doing 1. All right, but what we see here, centrioles, the aster, which is your spindle fibers that are now forming nicely. You've got the nuclear envelope. You've got the nucleolus, but the nucleolus is now starting to what we call, and I'm going to do this in yellow so you can see it nicely here, it starts to disappear. Okay, it doesn't go away, it just disappears. It moves right out of the process because it's not important. Then we have our chromosome pairs. Now look at it. It's a pair of chromatids, all right, there, but this is your chromosome. So this whole, and you can't see the yellow, so let's go here. This is one chromosome, and here's another one. And chromosomes are always in pairs. Why? We get one chromosome from the female and one chromosome from the male. And the two together are what we call diploid, and that is what we have. So you're always going to have an even number of chromosomes. So if you look at a diagram and it only has 3 chromosomes, you know, this diagram's wrong. It must have 2 or 4 or 6 or 8 or 10. It's always in pairs. It's the same as you can't wear one sock. You need 2 socks. Right, so chromosomes are always in pairs, and then you get your centromere, which is in the middle of a chromosome. And I think I've got like 1 minute left here, so I just want to show you this. Okay. Okay, I've got 3 minutes left. All right, let's do this one in yellow. So this is why we say you always have a chromosome pair. And the same with a plant, and the same with an animal cell. Makes no difference. Just flowers, different animal and plant cells will have different numbers of chromosomes. All right? And as humans, we have, do you know, 46 pairs of chromosomes, of which, 46 chromosomes, which is 23 pairs. 22 pairs are what we call somatic chromosomes. They are for your body and dictate what you are and what you're at. But if we look at the last one pair of chromosomes, that is your sex, those are your sex chromosomes. And you'll have XX if you're female or XY if you are male. But you've always got two chromosomes. They're always in a pair. And we say this pair is a homologous chromosome pair. Now the word homo means the same. Hetero means different. So homo is the same. Homologous, they are chromosomes that have the same genes on them. So this one will have a gene for eye color, this one will have a gene for eye color. This one will have for a funny shaped toenail, that there you'll have a funny shaped toenail. This you got from your mom and this you got from your dad. They are homologous pair of chromosomes because they have the same genes on them. Right, so people, you've got to remember it. Now, what's this chromosome made of? Well, this is, those two little legs are chromatids. Chromatids. So they're two. So that's one chromatid, that's the other one. And the two chromatids, which look like this. And there's another one here. They are joined together in the middle here. And this thing here is called, I'll be finished in one sec, the centriole. Right, that's your centriole. So what have we got? Your centromere. What am I doing? It's because they're telling me I'm
Don't panic, Kathy.
Centromere. So the two chromatids are held together in the middle by a centromere. And that, people, is incredibly important because when you get to meiosis, you need to know the difference. All right, Indy.
Okay, guys, if you have a look on the page, you'll see that I've posted all of the diagrams that you need. Download them onto your cell phone so you've got them. Mindsetters, we've run out of time for questions. If there is anything that you are struggling with, please paste it onto the page. Mindsetters help each other out. Grade 11s and grade 12s, I know if you're on the page, you're going to be able to help the grade 10s out. And you know that you'd want somebody to do it for you. Guys, remember next week we'll be wrapping up on cell division. Get your questions in early to make sure they get answered. And thank you, grade 10s, for such an amazing show. Grade 11s, don't forget you're up next. Bye, guys. See you next week.